record his presentation. So you should all have a message now that this is being recorded. Okay, so it's a pleasure to introduce Manuel Rodriguez. So he actually told me that he just arrived from Mexico, so he's a bit cold today. So let's see if we can get him heated up today. So Manuel is a research director, DR2, in the French CNRS. His research is oriented to the study of post-translational modification of the ubiquitin family and their impact in the regulation of physiological and pathological events. So he's working in collaboration with both public and private institutions to explore distinct pathologies, including cancer, inflammation, neurodegenerative and diseases and infections. So he's particularly interested in understanding the impact of these PTMs in the regulation of crucial cellular functions and how they are affected in diseases. So to this end, he have developed several molecular traps that capture these ubiquitin family-like modifications. And I'm sure we will hear more about these traps today. And these technologies, they can be used for detection, quantification, and also in high throughput screening. He's very active in coordinating various European research networks. So for example, the UB code. So with this, I will give the Zoom floor to you, Manuel.
Yes. Well, thank you very much for this invitation. It is a pleasure for me to share some of the experience we got when working with these tools that we call now molecular traps. It wasn't our intention to develop any technology at the beginning, as it happens in fundamental research. We were just exploring possibilities to study the ubiquitin proteome at basal conditions without any transfections, any artificialities, because we realized that when overexpressing or when including huge tags, the information we obtained was different. So then the necessity that we realized at that time was based in our observations on the overexpression of ligases that were at the same time the ubiquitinases. So then that was a very complex paradigm to explore using the technologies available at that time. So then what I will present today is a bit the summary of the experience over these last 10 years. And I will start with a small introduction for the not experts in the ubiquitin area, trying to justify the approaches we have developed and present some of the tools we are currently using. And then I will concentrate in 3 examples. One is the work we have performed in collaboration with GlaxoSmithKline that are based in Tres Cantos in Madrid to explore the role of ubiquitin in the regulation of Plasmodium falciparum infection. And then after I will continue to the study, we also started in Spain exploring the role of ubiquitin in the regulation of the response to the proteasome inhibitor bortezomib in mantle cell lymphoma cells. These 2 projects have in common one particularity, is that we started with this problematic that Plasmodium falciparum as mantle cell lymphoma develop resistance to proteasome inhibitors. And then that was the starting point of the 2 projects. And that's why I put them together today. And then finally, there were some similarities that we could observe in between the 2 completely different systems. And finally, the third part that I will finish with is the development of molecular traps to capture LC3 RABA gap proteins, which are involved in autophagy. So then, as you know, a protein homeostasis is regulated at many different levels, including the synthesis of the proteins, and then the regulation of the activity of the proteins, often mediated by distinct post-translational modifications, including phosphorylation, acetylation, and ubiquitin family members, that finally are able to connect with the different functions of the cell. Some of these post-translational modifications are also connected to the regulation of the protein stability and localization. And when those mechanisms fail, often pathology is observed, including multiple infections, inflammation, cancer, and neurodegeneration. For today's presentation, I mean, it is impossible to illustrate all the examples that we have been working with different partners. So I have decided to concentrate in the ones I have listed before, trying to go a bit deeper into the progress we have done in those problems, in those systems, and just to illustrate the potential applications and interesting that can be developed in this project when you use our molecular traps. So as you know, the conjugation of ubiquitin is mediated by a tious cascade of reactions, including 3 enzymes. Some of these enzymes are more important to provide specificity to the modification of the target proteins. However, all these enzymes represent targets for intervention for therapy, as it has been the case and published by several groups over the last years. So an important regulatory step also of this ubiquitination cascade is the reverse reaction mediated by the ubiquitinated enzymes that remove the ubiquitin chains and then proofread the proteins that will be degraded or not, or will be used to connect with different functions. The complexity of the system increases due to the fact that ubiquitin contains several active residues that allow the formation of ubiquitin chains. And 7 of these residues are lysines, but also the C-terminal, the N-terminal methionine can be used to form chains of ubiquitin. So then with that, we reach a complexity that was more important than initially suspected. So in which we can classify what we call typical chains and atypical chains. I think all these names that were given to didactically explain the problematic in reality is a very simplistic way to illustrate the potentiality to form different chains. Since often in one particular protein, there is more than one chain that is formed. So, but in the way the different chain types are conceived are typically associated to either proteasomal degradation for the chains formed by ubiquitin and the lysine Ki48 and K11. Typically, K63 chains are linked to DNA repair, signal transduction, and autophagy degradation. Other atypical chains are connected to many distinct functions, and it is difficult to group them in a generic function that they are regulated. I think in part this is due to the fact that we lack of tools to explore the relevance of those less abundant ubiquitin chains, and then generic functions cannot be established right now for most of these chains. So then with this, we reach to a complexity that is not only composed by the diversity of the ubiquitin chains, but also the participation of other ubiquitin-like proteins that can integrate ubiquitin chains, and then also by the capacity of some domains present in many proteins that work as receptors that are able to identify all this diversity of chains composed not only by ubiquitin, but also by ubiquitin chains, by ubiquitin-like proteins. So then in that way, these receptors contribute to connect with the different functions that are known to be regulated by ubiquitin or ubiquitin-like proteins. And as I said before, the interesting point from the pharmaceutical point of view is that many of these steps can be used to predict diseases or to target, to develop therapeutic approaches for these eventual target proteins. So then, given this complexity of the ubiquitin chains, many tools have been used over the years. Some of the most popular ones, including the tagging or ubiquitin-like proteins with distinct tags such as histidine, HA, FLAG, MIG. Almost all possible tags have been used. Many people use histidine tags because when you use histidine tags, you can prepare the cell extracts with guanidinium, so under denaturing conditions, which is very convenient to block the activity of the enzymes, including the deubiquitinating enzymes, and preserve the modified proteins for longer. So then other tags, including streptavidin, as you know, biotin and streptavidin have very high affinity, and then for some applications, this is very convenient. More recently, ubiquitin-specific antibodies have been developed, including those recognizing the double glycine motif that is located in the C-terminus of the ubiquitin and other ubiquitin-like proteins, facilitating the capture and the mass spectrometry analysis of the targets modified these ubiquitin-like family members. More recently, so then there is chain-specific nanobodies that have been developed by several groups, including us in participation, in collaboration with Hypergenics, but other groups have also developed nanobodies as well. Then also recently Affimers, and then in our case, and the purpose of this presentation is to introduce a bit what we have done to develop these tools that we call now molecular traps that are able to capture ubiquitin or ubiquitin-like proteins. The concept of molecular traps, in reality, it comes from observations of natural proteins that contain more than one binding domain. Typically, for those of you working with SUMO, you know that the SUMO-dependent ubiquitin ligase RNF4 contains 4 domains of 4 SIM domains, which increases the affinity for SUMO. So then this is very relevant for the ubiquitin ligase function of this enzyme. However, many other proteins contain multiple domains, such as RAD23, which contains several UBA domains. And based on these observations, we decided to artificially create proteins that contain multiple domains in order to increase the affinity and develop better purification tools. So then currently the tools that we have available are different versions of the ubiquitin traps, SUMO traps, net traps, and more recently, the autophagy traps that I will illustrate some examples during this presentation. So the prototype molecular traps were reported as tandem ubiquitin binding entities and were based in the multiplication of several ubiquitin binding domains of the UBA domains from the ubiquilin 1 protein and Ra23. And as you can see in this first experiment developed by Roland Jerpe, at that time PhD student in the laboratory, is that he used these traps to capture ubiquitinated I-kappa-B alpha from cells stimulated with TNF alpha, which induces the ubiquitination of I-kappa-B. And what you can see here is the capture of the two compared to single ubiquitin binding domains of the two different proteins that were used to develop the prototypes, ubiquilin and Ra23. And as you can see here is that adding 4 times the amount of UBA domain does not provide the same effect than the capture performed using the two, as you can see here. So then this suggests that the tandem repetition has additional properties to the trap, and this can also be observed when you capture total ubiquitinated proteins from the same experiments, as you can see in this Western blot in the bottom of this panel. One big advantage of these tools is that it has 2 particularities. Is that one is that interferes with the action of the deubiquitinating enzymes by molecular obstruction. And you can clearly see that here in these 2 experiments, in which we compare with the combination of chemical inhibitors, standard doses of these iodacetamide and N-methylmaleimide. And you can see here that the tubes do protect better ubiquitinated proteins compared to this chemical mix of the ubiquitinated proteins, even better when compared to a single UBA domain. This is more or less the same observation that we have done in this case, but in this other experiment in the right, we have done a time course experiment to see how long we could protect ubiquitinated proteins when working at room temperature, because we were interested in doing post-modification analysis, and for us it was important to be able to work at room temperature. So then, in addition to this interference with the ubiquitinated enzymes, we have also protection with from the action of the proteasome. As you can see here, we were comparing the proteasome inhibitor MG132 with the incubation of Ra23, Ra23, using either reticulocytes as a source of proteasome or purified 26 proteasomes, and using as a target protein the tumor suppressor protein P53. And you can see that the protection is quite similar to the one we obtained with the proteasome inhibitor MG132, this in vitro degradation assays. So then these characteristics of the tubes, the high affinity, the high specificity, because exclusively interacts directly with ubiquitinated proteins, allowed us to develop several projects and several applications, and I will illustrate some examples right now. So as I mentioned before, one of the examples I have decided to present today is the work we have done in collaboration with GlaxoSmithKline. This collaboration stopped in a couple of years ago, but they continue working on these aspects. We hope that now we will reestablish collaborations with them. And this work was performed essentially by Lidia Mata Cantero and other group members, and it was essentially published in 4 publications that are listed here. that I will try to illustrate a bit in this presentation. So then, first of all, we have established a protocol to isolate the ubiquitin proteome from different biological samples, and using as a control column a GST column, because the prototype troops were based in the GST fusion. So we developed affinity chromatography, and after elution of the samples, we sent to mass spectrometry analysis, and the information we obtained can be grouped, as I meant, as it listed here, in ubiquitin regulated functions, ubiquitin chains, and ubiquitin enzymes. So I won't present all the different sections of the information we obtain. I will just try to illustrate some of this information that we continue to explore, to give some, to try to understand the process that we were exploring, and also to try to see if there is any potential application from the knowledge extracted in the different, obtained in the different projects. So then for the Plasmodium falciparum project, we have used infected red blood cells. This was the first challenge Lydia was facing. It was trying to get cultures of Plasmodium falciparum in the different phases of the infection, in particular the ring stage, the trophozoite stage, and the schizoid phase, in a synchronous way. That means that when you have an infected culture, you have populations of red blood cells that are infected at different phases. So the first challenge for Lydia was trying to synchronize, to enrich the different phases of the infection, and trying to get clear conclusions from the data obtained from these settings. The second challenge Lydia was facing is that the fact that the red blood cells, as you know, are very enriched in hemoglobin. So hemoglobin was a nightmare for this particular project, because it's a very sticky protein, and then we were trying to see the way how could remove the hemoglobin that we were obtaining during the purifications. Finally, Lydia managed to establish this protocol, in which we still see some background, as you can see in the GST control column. However, we can clearly see differences, not only between the control column and the tube column, but also from the different stages of the infection. We were comparing with uninfected red blood cells and infected red blood cells in the different phases. So from the more than 1,000 proteins that we obtained from these, we identified from these, using this protocol, we try to understand how interpret this information. And something particularly interesting for this project is that it was the first time that we were analyzing 2 ubiquitin proteomes simultaneously from 2 different species, the Homo sapiens and Plasmodium falciparum. And the difficulty here is that since ubiquitin is very well conserved, the antibodies recognizing the ubiquitin of the 2 species were not able to distinguish between the 2 species. So we have set up conditions to separate the infected fraction from the non-infected fraction, and trying to get clear conclusions from these experiments. And as example, I would just cite one of these proteins that we have identified that GSK continue to work with. This is REX1, which is a protein that regulates the export of protein, and regulates also the remodeling of the erythrocyte and the virulence of Plasmodium falciparum. So something very interesting is that when we could detect REX1 in different phases of the infection, in particular in trophozoites, as you can see here, this is mass spectrometry data that was analyzed in collaboration with Rune Mattisen, help us a lot with the data interpretation. And as cellular molecular biologists, and we also like to confirm by Western blots these observations that you can see here. Mass spectrometry people say that mass spec confirm Western blot data, and but we also like to continue to see the information by Western blots, even if I understand the point of view. We are kind of more happy when we do see the correlation in between the two data, as it is the case in here. So you can clearly see that as detected by mass spectrometry, trophozoite, in the trophozoites, REX1 is heavily ubiquitinated compared to other phases of the infected red blood cells. And interestingly, REX1 belongs to this Mohrers-Cleft system that regulates a sorting compartment between the parasite and the erythrocyte membrane. And we have identified the different proteins that are here illustrated with a shadow. We identified all these proteins, and so the proteins that appear in blue are proteins from Homo sapiens, and then the proteins indicated in black are from Plasmodium falciparum. So then we see that there is a coordination of the functions that are in which the parasite takes advantage of the host machinery in order to continue progressing in the replication. And this could be better observed here because we have integrated this information in different ways. So then if you see the proteins, the total proteins we have identified from this study in the different phases of the we have integrated here. Here you see the total of almost 200, from which almost half and half belong to the Homo sapiens and Plasmodium falciparum. Something really interesting is that while the ubiquitin proteome in Homo sapiens was decreased during the progression of the infection, the ubiquitin of the Plasmodium falciparum was increased over the infection. And you can also see here by Western blot, when we separate the fractions from the Homo sapiens and from the Plasmodium falciparum infected fraction, you can clearly see that while the total ubiquitination on Homo sapiens decreases, there is an increasement in the ubiquitination of the Plasmodium falciparum proteome or ubiquitin proteome fraction. You can also observe exactly the same information here in these heat maps, in which Rooney helps us to integrate this information. You can clearly see that the ubiquitin proteasome system decreases over during the infection for the host, but increases during the Plasmodium falciparum infection for the parasite. Other processes also decrease, like for the host, the activity of some peptidases. However, DNA replication increases for the parasite. So not all the functions decrease in the parasite or in the host. There is several functions that are simply regulated in a completely different way. And as I said before, we went to check that by Western blots. There is only very few available antibodies from Plasmodium falciparum. You know, the problem when the diseases are considered as a cured disease, then there is less investment in regions and tools. However, now malaria is considered as an emergent, new emergent infection. So then since the weather conditions are changing in the world. So then we see more and more countries that they didn't have as much malaria. Now they are developing more. So then now they are investing a bit more in this type of diseases that were kind of almost forgotten diseases. So then we could confirm exactly the same observations, that the proteasome system from the parasite was increasing, while the ubiquitin proteasome system in the host was decreasing. So for this reason, we consider that tackling the proteasome could still be a good point to address in this project, and we decided to continue working on that. However, it was important to develop proteasome inhibitors that were not affecting the host, but only the parasite. And this is exactly the reason why we have started developing this project with GSK. So then we decided to go for a screening campaign. As you know, GSK has one of the most, the largest libraries and chemical compounds. So then we have not started with the 2 million compounds that they have. We started with the sub-library that was already characterized and active with malaria. So then it was 200,000 compounds. So then we developed two different types of assays. One homogeneous assay based in alpha ELISA, another heterogeneous assay based in Delphi assay. Both of the assays established by Lydia worked, and then we use one first screening to isolate most interesting compounds, and then we have as a confirmatory test the Delphi assay. So with that, we could identify some proteasome inhibitors, but also non-proteasome inhibitors molecules that still are on characterization by GSK. I will concentrate only in these proteasome inhibitors. Very quickly show you that they were affecting the proteasome activity of malaria and were not affecting the proteasome activity of the host. And the reason why was exclusive affecting the Plasmodium falciparum proteasome, it was because of the pocket that is allowing the binding of some of these compounds. In here, we are just illustrating this compound called GSK 737. All this information has been published, so then you can go and check if you are interested to know more about these molecules. But the interesting point is that we could demonstrate that these compounds were very effective in blocking the parasitemia. And for this reason, they are evolving, making an evolution of these first molecules to develop more specific and better solubility, more efficacy of the molecule for tackling malaria. So then this example was just to illustrate that our tools can be used to screen complex libraries. And we help in this project to reduce the number of molecules that can pass to clinical studies. So then hopefully they will reach this purpose at the end. Another very quick example that I have included here is to illustrate the project in which we were interested to understand the role of ubiquitin in the bortezomib-resistant mantle cell lymphoma cell. This project was started by Wendy Xolalpa, and then continue with Grégoire Quinet, and finally with the participation of Maria and Gonzalez in some of these aspects. So understanding proteasome inhibition is not an easy task, because, as you know, proteasome is involved in so many different functions that it was very difficult to predict which functions were the most affected. And certainly there are several functions that are affected, and we just decided to concentrate in only a few of them. So for this project, we started with a large collection of mantle cell lymphoma cell lines that were provided by Gael Rouet, which is also part of this consortium, Proteocure. He is based in the Joseph Carrera Leukemia Research Institute in Barcelona. So then he already had at that time cell lines that were naturally present and resistant to proteasome inhibitors. So these patient-derived cell lines. And he also had a couple of cell lines that he developed for acquired resistance of bortezomib. The advantage of using this couple of cell lines is that you have the parental cell line and the bortezomib resistant cell line. In this case, GECO, GBR, and Z132 and ZBR, these were the cell lines were used in particular for the data that I will present here and for the mass spectrometry analysis we have performed. In this case, I have also decided to concentrate in the functions affected. I will not develop the other two aspects. Hopefully in the near future, we will have the opportunity, either one of my students or myself, to present that in another meeting. So then, as we continue working with that, with René Mathisen, he helped us to integrate this information in these gene ontology-based heat maps to select the functions that were more enriched. We could also confirm using Ingenuity pathway analysis that the top 5 functions affected included the ubiquitin proteasome pathway, trans signaling, phagosome maturation, cell cycle control, and unfolded protein response. So 3 of these functions are directly related to protein degradation and stability. And for this reason, we have decided to concentrate in exploring this function. So the first thing we have observed is that while in the bortezomib resistant cell line, the autophagy lysosome system was enriched, the autophagy, the ubiquitin proteasome system was enriched better in the sensitive cell lines. So suggesting that there was a sort of crosstalk or compensatory mechanism that was established in between these two systems. So by that time, sorry, there was a webinar presented in the previous post-action Proteocure, in which we could hear Richard Riestra that he published a process that was reported in Arabidopsis thaliana, which was known as proteophagy, in which the proteasome was degraded by the autophagy lysosome system. And we have decided to explore this possibility. So the first thing we have done, of course, was to confirm the mass spectrometry data by Western blots, as we like to do it. So then we could confirm that not only proteasome subunits were decreased, but also autophagy proteins were increased in the bortezomib resistant cell line compared to parental cell line. Here I'm just presenting the data with the ZBR and Z132, but similar observations were observed in other couple of cell lines. So then when blocking the autophagy using different autophagy inhibitors such as baphlomycin or chloroquine that I'm not presenting here, we could observe that there was an accumulation of proteasome subunits in the bortezomib resistant cell lines, as you can see here in these black columns. And also there was an enrichment of the autophagy receptor P62. So we could confirm that it was an active proteophagy in these bortezomib resistant cell lines. And we also could demonstrate that the sensitivity to bortezomib was increased when we were combining distinct autophagy inhibitors to recover the sensitivity of these cell lines to bortezomib. One particular autophagy inhibitor is blocking the P62 autophagy receptor, bertoporfin, was providing the best recovery of sensitivity to bortezomib, and for this reason, we decided to concentrate in trying to understand the role of P62 in this process. So then something that it became clear is that P62 is playing an important role in the sensitivity to bortezomib, since when we silence P62, there is an effect on the proliferation levels in response to bortezomib, so suggesting that indeed P62 was playing an important role also in the sensitivity to this proteasome inhibitor. But something very interesting is that bertoporfin induces aggregates of P62, as you can see in here, and compared to the treatment that is done with bafilomycin, for instance. And there is also something very interesting is that you can see autophagy activation here as seen by the levels of LC3A and the lipidated form LC3B. That is observed when you block with bafilomycin, but you do not observe when you block with bertoporfin, because this is acting upstream the autophagy pathway. Even more interesting is that we could identify that P62 was playing an important role in the recruitment of the proteasome subunit RPN1. In this experiment, which is an IP experiment with the antibody P62, in which we compare the treatment of bertoporfin compared to the bafilomycin. So since bafilomycin is acting downstream the cascade, you accumulate these sort of protophagosomes, compared to the bertoporfin that is blocking upstream and does not allow the accumulation of the protophagosomes as when we do it with bafilomycin. So then the importance of P62 was further explored also in xenograft models of mantle cell lymphoma cells. In here, we have used REC1, which is the naturally resistant cell line. And you can see that the bertoporfin aggregates P62 in these tumors. And also that the treatment, combined treatment of bertoporfin and bortezomib reduces the tumor size in these animals, as indicated in these graphs. So then in conclusion, thanks to the use of this approach, we could identify that proteophagy, an important mechanism of crosstalk in between the ubiquitin proteasome system and the ubiquitin and the autophagy lysosome pathway was observed. There's still many questions to address to respond to many answers, to many questions that are still open. Including how this process is regulated, which are the sensors detecting the proteasome that is not active anymore, which is the role of ubiquitin in this process, and then obviously how can understand the impact in the different proteasome populations that we could detect in these cell lines that still present some proteasome activity despite of the fact that the proteasome activity is reduced. So then to further explore these, we have developed these, what we call the autophagy traps or LC3 traps. And this was also part of the work that was developed by Grégoire Quinet. So the principle is the same, is the multiplication of these Lear motifs, so LC3 interacting domains. So we developed these tools containing multiple repeats. We demonstrate that these tools could be used to explore processes regulated by autophagy and to capture the different ATGA proteins in different biological models, including C. elegans and also cerevisiae. So then we could demonstrate that these tools have been used to explore selective autophagy processes such as mitophagy. In this case, we have used cerevisiae to demonstrate that, but also human cell lines treated with distinct chemical inhibitors to activate mitophagy. So then, and finally, and the reason why we have created these tools, we could demonstrate that we could also isolate autophagosomes containing proteasomes. As you can see here, these are better captured with the trap base in the Lear domains enriched with tryptophan, but also we can observe LC3B, RPN1, RPN10, and also P62 that is also enriched in these proteophagosomes. So the questions remaining are very many. And for now, we are trying to focus our attention in the different ligases we have identified, trying to understand if those ligases could be sensing the connection in between the ubiquitin proteasome system and the autophagy lysosome pathway. We have several candidates, including some TRIM ligases that provide the interesting results. Hopefully, in the next future, we will be able to present you some of this data that is still ongoing process. So then in some, the distinct molecular traps that we have developed can be used for different purposes, including detection using different procedures. In here, these traps are labeled with biotin and can be used to detect in high microscopy resolution. So then ubiquitin related proteins, but the same application is currently under development for other traps. Of course, the mass spectrometry application that I have already present you, but also some other techniques, not only the high throughput techniques that I already mentioned, but also quantification of total ubiquitinated proteins and also protein microarrays that are very useful to detect as biomarkers of different diseases that we are working, and also to validate some of the potential drug targets that we are now tackling. So then, of course, this work over the years cannot be possible without the participation of many groups and many collaborators. That some of their faces and institutions are listed here. Not all of them, because the space is limited. And now I would like to thank also the past and actual members of the group, and obviously the funding agencies that have provided the different funding to continue developing here the many different projects we are implicated now. And thank you for your attention.
Okay, thank you, Manuel. It was a very nice overview of the, I guess, last 10 years of your research in your lab. I enjoyed very much the overview, and it was also a very nicely timed presentation. So is there any questions from the audience to Manuel? So maybe I can start with one question. With the LC3 traps, have you tried to look into other modifications that could be enriched by these traps with, for example, MS?
With the LC3 traps, actually, we have focused for the moment just in the detection of the different members of this LC3 GABA RAP family. We have done some work in yeast, and we have done also some mass spectrometry also in C. elegans. Actually, in C. elegans, we are also using this tool to distinguish autophagy-dependent, LC3-dependent autophagy events from the autophagy-independent events. But we have not, at the moment, explored the possibility of detecting, for instance, phosphorylation on the proteins that we are detecting right now.
Okay, thanks. So Nico have a question?
Yes, that was a very nice overview, Manuel. Can you hear me?
Yes, yes.
Yeah, okay, good. Like a principal question about the use of tubes, like since you are using their ubiquitin binding domains that the endogenous proteins also have, I guess that when you're pulling down the ubiquitin-related proteins, at the same time you will pull down also proteins that have ubiquitin binding domains and bind to the ubiquitin-related proteins.
Sure, sure.
Do you know to what extent that complicates like the interpretation of the data, that you have proteins that are not ubiquitin-related with ubiquitin, because there are a lot of ubiquitin binding proteins?
Sure. So then we definitely cannot exclude, like, for instance, we have detected when we use the tube based on RAD23, we have detected RAD23. Obviously, we cannot exclude that this also, it comes from complexes containing RAD23 that are not necessarily part of the UBA domain, right? However,
I think that RAD23 is not a good example because RAD23 is ubiquitin-related actually.
Yes, sure. So that's the thing, is that many of these proteins are also ubiquitin-related. So then I don't know if we will be able to exclude the possibility to pull down these other proteins containing ubiquitin binding domains. So what we know is that the affinity of these artificial tools is much higher than the natural ubiquitin binding domains.
Yeah, I was wondering that because you use like 4 of those ubiquitin binding domains, which is more than what a natural proteins have.
Yes, yes.
So
yes.
the affinity should be higher. But have you tried with more stringent washing that you can get rid of the ubiquitin binding proteins?
Yes, I mean, we at some point were doing mass spec by fractionating the different let's say fractions of the proteome by using different elution conditions. But then it's not something like a black and white. So then while you increase more the stringency, obviously you obtain more ubiquitinated proteins, but in the first washes, we were also obtaining some ubiquitin proteome and interactome, so what we call interactome. So then after all, we realized that the interesting of this tool compared to other tools like histidine-related versions of ubiquitin is that you can purify also the interactome. And that is also connecting with other functions. So then gives you kind of clues that which functions you can explore. Because the whole thing is integrated in complexes. We don't destroy the complexes. This is done under non-denaturing conditions. And then I think thanks to that, we could connect with the different functions we finally have managed to integrate.
Yeah, okay. And yeah, I noticed that people sometimes refer to it as the ubiquitin that they are pulling down, but I think you're pulling down more than just the ubiquitin.
Yeah, sure, sure.
Yeah.
Which is, it is true. If you want to get only the ubiquitin, so then better to use one of these other strategies, you know. So then I don't know if, like, for instance, I think all the strategies that I have listed have positive and negative aspects. You know, the anti-GG antibodies, they pull down more than just ubiquitin, and you know, and the ubiquitin antibodies, they do not protect like the tubes. The tubes have this advantage that and protect ubiquitin-related proteins. So I think all the tools when we use them in combination, including the ones that we have recently developed, for instance, the nanobodies, these are very good, but they do not offer protection. So then you really need to work with conditions in which you actually protect your sample using all possible sort of inhibitors, proteasome inhibitors, and DOPS inhibitors, and only under these conditions you manage to get some information. So then I think with the whole set of tools, we are now able to address many questions from different type of perspectives.
Okay, thank you.
And Malte?
Yeah, thanks a lot for this fantastic overview and the really clear talk and sharing all your insights here. So Nico basically just asked the question that I wanted to ask, so I'm just going to ask a follow-up question on the cellular screening that you've done. So I understand it correctly, this was a detection based on a ubiquitin antibody. That was the second readout, so you would read out global ubiquitination. And that is
yes. So you mean in the high-throughput screenings?
Exactly, in the alpha LISA, for example.
In the alpha LISA, so then we have used the ubiquitin trap as tool for detection, and then the sandwich was completed with a ubiquitin antibody.
So I'm wondering, have you does this give you a lot of signal? And if so, have you tried dialing this down to an antibody that detects a specific protein so that you don't measure sort of global ubiquitination, but sort of specific ubiquitination?
I think if you have a good antibody for a specific protein, this is possible. We have not done it.
All right.
But certainly it is possible. I mean, technically it's possible. And it's just you need to have a good antibody.
Thanks a lot.
Okay, Mustafa.
Hello, and
Yes.
thank you so much for your nice presentation. But just as a curiosity, I have a question in case of the mantle and the generation of resistant cells to the proteasome. It's just maybe a simple question for you, but I want to know that why you chose the mantle cell lymphoma to generate the model of the proteasome resistant cells. Why you didn't choose cells with more secretory activity and protein homeostasis activity like multiple myeloma cell lines?
Okay. We have done some work with multiple myeloma. However, we have started with mantle cell lymphoma because getting lymphoma cell lines is more easy for access than the multiple myeloma. When you have access to the different collections, because we didn't want to stop only into the analysis of cell cultures. We wanted to go to analysis of the patients, what happens in reality. And then for us, it was more easy access to get the mantle cell lymphoma samples from patients. This is not anymore the case now. We also have access to multiple myeloma, and some of these observations we have done in mantle cell lymphoma are somehow confirmed in multiple myeloma, but not all of them. Maybe the complexity of the disease makes that there's not a straightforward comparison between the two cell lines. To tell you the truth, I mean, even in between the cell lines that we have used and the samples from patients in mantle cell lymphoma, the response is not necessarily the same. And this is because you have multiple somatic mutations in the different patients. So the response is not exactly the same. However, some of the features are preserved. For instance, the activation of proteophagy when there is a resistance to proteasome inhibitors.
Yes, thank you. Thank you so much.
Okay, Thanks. Are there more questions? Otherwise, I will allow myself to ask a last question, and that is, are you still working on traps, and what will be your next trap?
Well, actually, if you see, there is a sort of continuity in the work we have developed over the years. So then we are driven by the questions we are addressing. So for instance, the last traps we have developed of autophagy traps or LCT traps is because we identify proteophagy, you know, so then we wanted to better understand how the process was regulated. So for this reason, we developed these traps. So then I think we follow the natural continuity of the projects, and we don't know right now what are these the future questions will be. I think we will continue using the strategy because it works, but I cannot tell you. It depends on the results.
Okay, thank you very much, and thanks for everyone for attending today. So next week we have again a speaker. It will be Camilla, and I hope to see you all there.